Today, our topic will be about enzymes, the biological catalysts. Enzymes are organic compounds that act as catalysts for a biochemical reaction. So, as catalysts, what they do mainly is to speed up the reaction. These are specialized proteins with fascinating precision and selectivity catalyzing biochemical reactions. Enzymes cause cellular reactions to occur faster, so that is their main function. Most are globular proteins, and enzymes can be proteins and RNAs. Though most enzymes are found to be proteins, there are some enzymes that are of nucleic acids in nature. Now, before, scientists thought that enzymes are purely proteins because the first discovered and studied enzymes are the digestive enzymes like pepsin, trypsin, and chymotrypsin, which are protein in nature. So scientists thought that all enzymes are proteins. But later on, when many enzymes are discovered, they discovered that some are made up of nucleic acids. But in this chapter, we will be focusing on the characteristics of enzymes as they are proteins. pH, temperature, or other protein denaturation affect enzyme activity. Enzymes, as proteins, are also very large molecules, and they are regulated by other substances in the cell. Okay, so let us talk about enzyme structure. If we would be classifying enzymes based on their structure, we can classify them as simple and conjugated enzymes, just like how we classified proteins. Well, we have simple proteins and we also have conjugated proteins. So for enzymes, we also have simple and conjugated. Simple enzymes, as you can see, are enzymes composed only of protein. So you have this protein portion, which is made up of amino acids. While conjugated enzymes are those enzymes that, in addition to the protein portion or to the protein part, has a non-protein part which can either be a cofactor or a coenzyme, which is also a cofactor. Now, cofactors are the non-protein part of a conjugated enzyme. Now, this protein portion, which is the inactive form of conjugated enzyme, is what we call the apoenzyme. Now, when we combine apoenzyme, or when the apoenzyme is activated by a cofactor, now we have a holoenzyme, which is the biochemically active conjugated enzyme, which is produced from a combination of an apoenzyme and a cofactor. Now, why do apoenzymes need cofactors? Cofactors, their function is that they provide additional chemically reactive functional groups besides those that are already present in the side chains of the amino acids of the apoenzymes. There are two broad categories of cofactors. We have the metal ion cofactors and the coenzymes. When we say metal ion cofactors, so these are zinc, magnesium, iron, and copper ions as an example. And these metal ions are supplied to the human body through dietary mineral intake. So almost any type of diet will provide adequate amounts of this needed metallic cofactors because we need them in very small amounts. Now, when we say coenzymes, most often one of the building blocks for enzymes would be a vitamin B or a vitamin B derivative. So coenzymes are small organic molecules that serve as cofactors in a conjugated enzymes. Now, when we think about cofactors, these substances are permanently bound through a covalent bond to the amino acid portion of an enzyme. Now, when these covalent bonds are broken, it would deactivate the enzyme. Metal ions and many, but not all, coenzymes are permanently attached parts of an enzyme. This permanent attachment of the cofactors is actually not an absolute requirement for a coenzyme to be an active part of an enzyme. Sometimes the coenzymes, they temporarily bind to the apoenzyme or the amino acid portion of an enzyme at the time it is needed, and then when it is not needed anymore, it would be released after the reaction has taken place. If we will be talking about the enzyme structure, we would be talking about two very important substances. First is the substrate. So the substrate is the reactant in an enzyme catalyzed reaction. Now the active site is the relatively small part of an enzyme, which is the one that is actually involved in catalysis, or what we call the catalytic site. On the other hand, an enzyme substrate complex is the intermediate reaction species that is formed when a substrate binds to an active site. So as you can see from here, so we have here the substrate. When it attaches or forms a covalent bond with an active site, we form what we call an ES complex, or an enzyme substrate complex. Now always think that this enzyme substrate complex is a temporary or an intermediate reaction species that after some time, which is usually very fast, would be converted into products. Now always remember that enzyme is a substance that is involved in the reaction, in a sense that it has the place where the reaction or the conversion of the substrate into products take place. But always remember that enzymes are not consumed in the reaction. So their main purpose is just to catalyze or to speed up the rate of the reaction. Nomenclature and classification of enzymes. Enzymes can be grouped into 6 major classes. But before we group them into 6 major classes, let us talk about the 3 important aspects of the enzyme naming process. First, most enzymes, or almost all enzymes, would end in the suffix ase or ase, which will identify that a substance is an enzyme. So for example, we have the urease, sucrase, lactase, and lipase enzymes. All of these are enzymes, and if you will notice, all of them end with the suffix ase. Now, the suffix in or in is still found in the digestive enzymes, which are some of the first enzymes that are studied. So we have trypsin, chymotrypsin, and pepsin. Next, the type of reaction catalyzed by an enzyme is often noted with a prefix. So for example, we have oxidase and hydrolase, so it ends in ase. But if you would notice in their prefixes, oxidase or oxy and then hydro. So from this, we can already determine the type of the reaction that this enzyme catalyzes. So the name of an enzyme would give us 2 information. The first is the type of reaction that it catalyzes. Third, it also gives us what the identity of the substrate is. So the identity of a substrate is often noted in addition to the type of reaction. So for example, we have glucose oxidase. So from the name of the enzyme, we would know that the substrate is glucose and that the enzyme catalyzes an oxidation reaction. So we have pyruvate carboxylase and succinate dehydrogenase. Okay, so these are the 6 major classes of enzyme. We have the oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. So let us take a look into each of these 6 major classes in details. First, we have the oxidoreductases. An oxidoreductase is an enzyme that catalyzes an oxidation-reduction reaction or a redox reaction. Now, oxidation-reduction reactions or redox reactions are not independent processes. Always remember that these two are linked processes that must take place together or that must occur together, meaning that if there is a substance that would be oxidized, there must be another substance that must be reduced, meaning a substance would not be oxidized unless another substance would be reduced. So, an oxidoreductase enzyme requires a substance, which is usually a coenzyme, that will be oxidized or reduced as the substrate is reduced or oxidized. So, from the example given, we can see that our substrate is lactate and our oxidoreductase enzyme is lactate dehydrogenase. So this enzyme removes hydrogen atoms from a molecule, which here is our substrate lactate. So if you can still remember from your organic chemistry or in the study of organic reactions in your organic chemistry, whenever you see dehydrogenation reaction, it would mean the removal of hydrogen atoms. So in here, our coenzyme is NAD+, and since this is an oxidation reaction, then lactate is oxidized, and therefore NAD+ would be reduced. So our product would be converted into pyruvate, wherein what has taken place is that in the lactate molecule, 2 hydrogen atoms were removed, and this caused the formation of a carbon to oxygen double bond. Now, the 2 hydrogen atoms that were removed were accepted by our NAD+, which caused it to become reduced, and the other hydrogen atom was donated into the cellular solutions. Now, in organic redox reactions, there are some things observed that take place which indicates that a reduction and oxidation reaction has taken place. So for this instance, our example, lactate was oxidized into pyruvate, and if you will notice, there is an increase in the number of carbon to oxygen bonds. So from carbon to oxygen single bond, it was now converted into a carbon to oxygen double bond. And together with that, if that is an oxidation reaction, then there would be a decrease in the number of hydrogen atoms or the number of carbon to hydrogen bonds. Now, the reverse of it, if the reduction reaction would take place, there would be an increase in the number of carbon to hydrogen bonds and a decrease in the number of carbon to oxygen bonds. So that would help you to determine whether a redox reaction has taken place. Next, we have the transferase enzyme. So this is an enzyme that catalyzes the transfer of a functional group from one molecule to another. Now, the transferase enzyme can be subdivided into two. So the two major subtypes are the transaminases and kinases. Now, for our example, we have here glucose reacted with an ATP molecule. And our enzyme in here is hexokinase, which is an example of a kinase enzyme. And after the reaction, ATP was converted into ADP, and our glucose has now accepted a phosphate group, or what we form a phosphorylated product, or a product which contains an additional phosphate group. So in here, what has taken place is that one phosphate group coming from our ATP is donated or transferred into the glucose molecule. Third, we have the hydrolase enzyme. This is an enzyme that catalyzes a hydrolysis reaction in which the addition of a water molecule to a bond causes the bond to break. Hydrolysis reactions are essential to digestion, and if you can remember, we have already discussed several hydrolysis reactions, the hydrolysis of carbohydrates and the hydrolysis of proteins and the hydrolysis of lipids as well. Now, hydrolases are those enzymes that are involved in the breaking down of the glycosidic linkages for monosaccharides, for disaccharides and polysaccharides. As you can see from our example, we have here the disaccharide maltose. So this glycosidic linkage is hydrolyzed by water, okay, in the presence of the maltase enzyme. So when this glycosidic linkage is broken down, our disaccharide is now converted into its monosaccharide components. Now for the hydrolysis of carbohydrates, so we have the carbohydrases, so meaning maltase is an example of a carbohydrase subtype of hydrolases. For the peptide linkages in proteins, we collectively call them as proteases. And for the ester linkages in the tags or the triacyl glycerols, we collectively call them as lipases. Now, an important component of hydrolysis reactions is the presence of the water molecule. Fourth, we have the Lyase enzyme. So this is an enzyme that catalyzes the addition of a group to a double bond or the removal of a group to form a double bond in a manner that does not involve hydrolysis or oxidation. So in our oxidoreductase enzyme, so these are enzymes that would catalyze the oxidation and reduction. And if you can remember, we would know that a redox reaction has taken place if there is an increase in carbon to oxygen bond and carbon to hydrogen bond. Now, for the Lyase enzyme, what has taken place, for example, in this reaction, fumarate, which contains a carbon to carbon double bond or an alkene group, is reacted with water. Okay, so this is an example of a condensation reaction wherein fumarate and water combines to form one product. Our enzyme or our Lyase enzyme in here is fumarase. So as a result, we form one alcohol product. So in organic reactions, whenever we hydrate alkenes or add water to alkenes, it would be converted into alcohol. And this is not an example of a redox reaction. So this is simply hydration reaction. So the type of Lyase enzyme involved is actually a hydratase. So hydratases are those that affects the addition of the components of water to a double bond. The hydratase would be the reverse, wherein the removal of the components of water to form a double bond is observed. Our fifth major class of enzyme is the isomerase enzyme. So this is an enzyme that catalyzes the isomerization or the rearrangement of atoms of a substrate in a reaction, converting it into a molecule isomeric with itself. Now take note that for isomerases or enzymes that belong to isomerase, they catalyze reactions wherein there is only one reactant and one product in reactions where isomerases are operative. So if you will notice in this isomerization reaction, initially we have the substrate 3-phosphoglycerate, wherein the phosphate group is on carbon number 3 and the alcohol group is on carbon number 2. Now after the isomerization reaction, there would be a rearrangement in the position of the phosphate group and the alcohol group. Sixth are the ligases. So these are enzymes that catalyze the bonding together of two molecules into one with the participation of ATP. Now, ligase enzymes would catalyze the condensation of two reactants to form one product, okay, in the presence of ATP. So here we have carbon dioxide added to the substrate pyruvate to form one product. ATP involvement is a requirement to such reactions because these reactions are generally energetically unfavorable. So they would require the simultaneous input of energy obtained by a hydrolysis reaction wherein ATP can be converted into ADP. So within each of these 6 main classes of enzymes, there are enzyme subclasses found in table 21-1 of your textbook. Okay, enzymatic browning. So, a commonly observed enzyme-influenced phenomenon that takes place outside the human body is the discoloration or the browning that occurs when freshly cut fruits and vegetables become exposed to air, even for such a short period of time. So, for example, we have here apples and potatoes which are peeled and cut. So, after being exposed to air, we notice that there would be a browning that would, or a discoloration that would take place. Now, the enzyme involved in here is phenolase, which is an oxidoreductase enzyme, which is present in our food and involved in the browning of some fruits and vegetables. Now, the substrates for the phenolase enzyme are phenolic compounds, which are present in the tissues of the fruits and vegetables. Now, in the chemical reaction, this phenolase enzyme hydroxylates phenol derivatives to catechol derivatives, and then afterwards this catechol derivatives, which is alcohol, would be converted into a ketone or the orthobenzoquinone derivatives. Now, the orthobenzoquinone derivative will now enter afterwards a series of reactions which will produce the unwanted brown discolorations. Now, the orthobenzoquinone derivative formation, remember, is both enzyme and oxygen dependent. However, once the quinone derivatives have been formed, the following reactions that will take place will now be independent of the phenolase enzyme and the presence of oxygen. Now, several ways can be done to prevent or to slow down enzymatic browning from happening. So, what we usually do is that we immerse the food in cold water to slow down the browning process. Now, the reason for this is that the lower temperature would decrease enzyme activity, and water will actually limit the enzyme's access to oxygen. So, a long-used method for preventing browning from happening would involve lemon juice or calamansi. So, the phenolase works very slowly in acidic environment or at a very low pH, which would be created by the lemon juice's presence. So, in addition to this, the vitamin C present in lemon juice or calamansi will function as an antioxidant. So, we learned that antioxidants would be more easily oxidized than other substances. So, here, vitamin C would be more easily oxidized than the phenolic derivative or the phenolic derived compounds, and its oxidation would produce products that are colorless. Okay, next we have models of enzyme action. So we will be discussing two. We have the lock and key model and the induced fit model. So the lock and key model, the active site has, as you can see from the representation on the screen, the active site has a fixed, rigid geometrical conformation. So meaning only substrates that have a complementary geometry or shape will be accommodated at the active site. However, this shows an inflexible interaction or shows that the lock and key model is too restrictive for the action of many enzymes. So, according to experimental evidences, many enzymes actually have flexibility in their shapes, which is not portrayed by the lock and key model. So that would be its limitation. So most enzymes are not rigid and static. So there is constant change in their shape actually. So a good analogy would be the changes that occur, for example, in the shape of a glove when a hand is inserted to it, right? So the name induced fit model is termed so for the induced small change of the active site of an enzyme such that the substrate can achieve optimal fit. So most enzymes are actually modeled by the induced fit model. So the enzyme active site, as you can see from here, is not exactly complementary in shape to that of the substrate, but it adapts or it is flexible enough that it can adapt to its shape. So in the induced fit mode, both the substrate enzyme's active site undergoes conformational changes until the substrate is fully attached to the enzyme, so it can be further converted into products. Enzyme specificity. Now, this is the extent to which an enzyme's activity is restricted to a specific substrate, a specific group of substrates, a specific type of chemical bond, or a specific type of chemical reaction. Remember that enzymes have active sites, and as you can see from the two models of enzyme action, as portrayed by the lock and key model and the induced fit model, enzymes are exhibiting specificity or selectivity. Now, the degree of enzyme specificity is actually determined by the geometry of the active site. So, some active sites accommodate only one particular type of compound, while others can accommodate a family of closely related compounds. Now, we have four types of enzyme specificity. First is absolute specificity. Second is the group specificity. Third is the linkage specificity. And fourth is the stereochemical specificity. So when we say absolute specificity, this is the most restrictive out of the four, or among the four. Enzymes exhibiting absolute specificity will catalyze only one reaction and only one type of substrate. Next, we have the group specificity. So, enzymes exhibiting this type of specificity will act only on molecules that have a specific functional group. So, for example, an enzyme exhibiting group specificity, for example, is only specific for those substrates containing alcohol functional groups. So meaning, if a substance or a substrate which has no alcohol group comes, it will not be accommodated in the active site of an enzyme which has a group specificity. Next is the linkage specificity. So enzymes will act on a particular type of chemical bond irrespective of the molecular structure. So for example, enzymes, the carbohydrases. Carbohydrases are those enzymes catalyzing the hydrolysis of the glycosidic linkage. So it will be specific, for example, for carbohydrates because they have the glycosidic linkages. And last, we have the stereochemical specificity. Enzyme will only act on a particular stereoisomer. So for carbohydrates, for example, most enzymes in our body would be inclined to carbohydrates that have the D designation or that are right-handed. Or for amino acids, our enzymes in our body are specific only for the L amino acids. Factors affecting enzyme activity. First, what is enzyme activity? Enzyme activity is the measure of the rate at which an enzyme converts substrate to products in a biochemical reaction. So it is a measure of how fast enzyme would be able to convert substrates into products. Now, there are four factors affecting enzyme activity that will be discussed. And first is temperature. So we can define temperature as the measure of the kinetic energy or energy of motion of molecules. At higher temperatures, molecules move much faster and collide more frequently. So this concept applies to collisions between substrate molecules and enzymes. As the temperature of an enzymatically catalyzed reaction increases, as you can see from here in the graph, the rate of reaction also increases. Now, optimum temperature is the temperature at which an enzyme exhibits maximum activity. So as you can see from here, as the temperature increases and then until it reaches its optimum temperature, the reaction rate also increases. However, when temperature increases beyond the optimum temperature, the increased energy would also begin to slow down the reaction rate because high temperatures would disrupt the tertiary structure of the enzyme and denaturation will take place, slowing down the enzyme or completely inactivating the enzyme. So for human enzymes, the optimum temperature is around 37 degrees Celsius or our normal body temperature. A person who has a fever where body core temperature exceeds or goes beyond 40 degrees Celsius can be in a life-threatening situation. So the reason for this is because this high temperature is sufficient enough to initiate enzyme denaturation. So when enzymes are denatured, they lose their function, and the loss of function of the critical enzymes in our body, particularly, for example, those in our central nervous system, can result in dysfunction sufficient to be fatal or to cause death. So the destroying effect of high temperatures on bacterial enzymes is actually the principle that is being used in a hospital setting to sterilize their medical instruments and laundry. Next factor is pH. Most enzymes exhibit maximum activity over a very narrow pH range. Optimum pH is the pH at which an enzyme exhibits maximum activity, and our normal physiological pH range would be about 7 to 7.5. However, there are some notable exceptions to this generalization, like our digestive enzymes pepsin and trypsin. So pepsin is the enzyme that is active in our stomach, and we know that our environment or the pH in the stomach is very acidic. So pepsin works best at a very acidic pH, which is about 2, while trypsin operates in the small intestine and it functions best at a pH of 8. A variation from normal pH can also affect substrates, so it can cause either protonation or deprotonation of the amino acid side chains on the substrate or of groups on the substrate, and the interaction between the altered substrate and the enzyme active site will be less efficient than normal or may even be impossible. So the pickled vegetables, like for example here in the Philippines we have achara, in western part of the globe they have sauerkraut, in Korea they have kimchi. So these pickled vegetables, we know that they do not readily undergo spoilage. So the reason is because of the acidic conditions during their preparation. So the acidity of these pickled foods actually significantly reduce the enzymatic activity of any microorganisms present. Now, in our stomach in our stomach, we have the H. pylori bacteria or the Helicobacter pylori, which are known to be the cause of most stomach ulcers. So they have an interesting adaptation to the acidic environment of the stomach, which allows them to grow in number. So you can read more on this in the chemical connections box in 21-B of the chapter on enzymes. Third, we have substrate concentration. So enzyme activity, as you can see from the graph, increases up to a certain substrate concentration and after that would remain constant. When the concentration of an enzyme is kept constant and the concentration of the substrate is increased, the enzyme activity becomes like the one shown in this graph in figure 21-8. So the activity pattern is what we call a saturation curve, meaning the enzyme activity increases up to a certain substrate concentration and remains constant from then on. As you can see from the graph, as substrate concentration increases, a certain point is eventually reached where enzymes' capabilities are used to their maximum extent. And then after that, the rate will remain constant from that point. As substrate concentration increases, enzymatic activity also increases. So from here, you will notice that the rate or the ratio between the substrate and the enzyme is low. So we have low concentration of substrate and high concentration of enzymes. But if we increase the concentration of substrate, the activity of the enzyme also increases. So each substrate in here occupies an enzyme active site for only a finite amount of time, and the products must leave the site before the cycle can be repeated. So once the substrate attaches to the enzyme's active site, forming an enzyme substrate complex, it will be converted into products. And once converted into products, it will leave the active site, and the active site would now be available for the next substrate. Now, when each molecule is working at full capacity, the incoming substrate molecules must wait for their turn for an empty active site. So at this point, the enzyme is said to be under saturation conditions. Turnover number is the number of substrate molecules transformed per minute by one molecule of enzyme under optimum conditions of temperature, pH, and saturation. Now, this table shows the turnover numbers for selected enzymes. Some enzymes have a much faster mode of operation compared to others. Last factor affecting enzyme activity would be enzyme concentration. In general, the concentration of substrate in a reaction is much higher than that of the enzyme. This is because enzymes are not consumed in the reaction, so the cells would usually keep the number of enzymes or the concentration of enzymes low compared to the number of substrates. This is efficient for the cell because it would prevent the cells from using a lot of energy for synthesizing and maintaining a large workforce of enzyme molecules. Now, if the amount of substrate present is kept constant and the concentration of enzyme is increased, the reaction rate increases because more substrate would now be accommodated in a given amount of time since the concentration of enzyme has been increased. Extremophiles and extremozymes. Extremophiles are microorganisms that thrives in extreme environments which humans and most other forms of life could not survive. So on the upper left part of the screen, we have an example of a very small extremophilic animal, which we call a tardigrade or a water bear, which is a very tiny aquatic invertebrate. So this tardigrade has the capability to live in a vast range of altitudes, depths, salinities, and temperature ranges, which for us humans, we cannot be able to survive. Extremophile environments include this example, yung hydrothermal areas of the Yellowstone National Park and hydrothermal vents on the ocean floor where temperatures and pressures can be extremely high. The ability of extremophiles to survive under such harsh conditions is related to the amino acid sequences present in their enzymes. So extremophiles have enzymes called extremozymes, microbial enzymes active at conditions that would inactivate human enzymes as well as enzymes present in other types of higher organisms. Some examples of extremophiles, we have acidophiles, alkaliphiles, halophiles, hyperthermophiles, those that can withstand very high temperatures, piezophiles for a very high pressure, xerophiles for extremely dry conditions, and cryophiles, those that can withstand very low temperatures. Okay, now let's talk about enzyme inhibition. Enzymes, we know, are required for most, if not all, processes required for life or biological processes. However, even if that is the case, enzymes still need to be tightly regulated because, as we can see from before, in enzyme concentration, remember, Usually, the concentration of enzymes are usually kept into low to um, conserve energy utilized by the cell. So, enzymes need to be tightly regulated to make sure that levels of the product do not raise to undesired levels. So, this would be accomplished by enzyme inhibition. An enzyme inhibitor is a molecule that slows or stops the normal catalytic function of an enzyme by binding to it. So, in this section, we will consider three models or three modes by which inhibition takes place. So, we will talk about reversible competitive inhibition, reversible non-competitive inhibition, and irreversible inhibition. In a normal reaction, the substrate will bind to an enzyme through the active site to form an enzyme-substrate complex. However, what enzyme inhibitors do is that they will prevent the formation of this enzyme-substrate complex, and therefore, as a result, will prevent the formation of a product. First, we have the reversible competitive inhibition. Competitive enzyme inhibitor is a molecule that sufficiently resembles an enzyme substrate in shape and charge distribution that it can compete with the substrate for occupancy of the enzyme's active site. So when a competitive inhibitor binds to an enzyme active site, the inhibitor remains unchanged. But yung physical presence at the site will prevent a normal substrate molecule from occupying the site, which will result in a decrease in enzyme activity because no substrates can be able to attach to the active site, so no substrates will be converted into products. The formation of an enzyme competitive inhibitor complex is actually a reversible process, so it's not permanent. The reason for this is because that complex, this competitive inhibitor enzyme active site complex, is maintained by weak interactions that can easily be broken down. So with time, which is usually just a fraction of a second, this complex will break up. And now, the active site will now be available once again for a new occupant. So after that, the substrate and the inhibitor will compete again for the enzyme active site. So take note that the portions of these two molecules that will bind to the active site have the same shape or geometry. So if you will notice, here we have the normal substrate and here we have the competitive inhibitor. Now the portion that attaches to the active site are both similar in geometry. However, the difference is that the shape of the normal substrate and the competitive inhibitor, yung overall shape, are actually different. So, the two molecules differ in overall shape. So, it is because of this difference that the substrate is the only one that will react at the active site while the inhibitor will not. If inhibitor concentration is greater than the substrate concentration, then the inhibitor will dominate the occupancy process and vice versa. Some examples of reversible competitive inhibitors are the antihistamines that we intake. So antihistamines are competitive inhibitors of histidine decarboxylation. So that is the enzymatic reaction that will convert histidine to histamine. Another one is ethanol for methanol poisoning. So ethanol is actually used as treatment in case of methanol poisoning. Ethanol competes effectively with methanol as an alternative substrate for the alcohol dehydrogenase enzyme, which catalyzes the formation or the conversion of methanol into acetaldehyde or formaldehyde, which is very poisonous for us. So what it does is that it would slow down the formation of formaldehyde because it competes with the active site. So it will lessen the danger while the kidneys will filter out the methanol to be excreted harmlessly in our urine. Second, we have reversible non-competitive inhibition. So a non-competitive enzyme inhibitor is a molecule that decreases enzyme activity by binding to a site on an enzyme other than the active site. So the difference between competitive inhibitors and non-competitive inhibitors is that the non-competitive inhibitor look for another spot in the enzyme where it can attach. Now, once this non-competitive inhibitor attaches to that site, so which we call as either the regulatory site or the allosteric site, it would cause a change in the conformation of the enzyme, and it would affect the shape of the active site, thus not being available for the substrate to attach. So the allosteric site or the regulatory site is the other site where the non-competitive enzyme inhibitor will bind. Now, the binding of the inhibitor to the allosteric site will cause a change in shape to the enzyme's active site. So, as the inhibitor is not in direct competition with the substrate, increasing substrate levels cannot stop or mitigate the inhibitor's effect. However, lowering the concentration of a non-competitive inhibitor will sufficiently free up many enzymes' active site, which can then return to normal activity. And also, similarly with competitive enzyme inhibitors, the binding of the non-competitive inhibitors to the allosteric site is not permanent as well. Examples are heavy metal ions like lead 2, silver, and mercury 2 ions, which cause heavy metal ion poisoning. So these heavy metal ions will bind to the sulfhydryl groups located away from the active site. So the heavy metal ions will form metal sulfide linkages, which will cause a disruption in the secondary and the tertiary structure of the enzyme. Now, to treat heavy metal ion poisoning, we can use another non-competitive inhibitor, which is the BAL or the British anti-Lewisite, or specifically the dimercaprol. Dimercaprol is used as an antidote for heavy metal poisoning. The heavy metal acts as an enzyme poison by reacting with a sulfhydryl group, remember? So the BAL or the British anti-Lewisite, dimercaprol, has also several sulfhydryl groups with which the heavy metal ions can attach to or can react, so thereby their poisonous effects are reduced. Third, we have the irreversible inhibition. Irreversible enzyme inhibitors are molecules that inactivates enzymes by forming a strong covalent bond to an amino acid side chain group at the enzyme's active site. So the difference between the reversible inhibitors and the irreversible inhibitor is that irreversible inhibitor competes with a substrate by attaching first to the active site. But the difference with a competitive inhibitor is that irreversible inhibitors, they will not have the exact same shape of the substrate that attaches to the active site. However, they can form permanent, strong covalent bonds to the amino acid side chains present in the enzyme's active site. So some examples of this are the nerve gases and the organophosphate insecticides. So since these covalent changes are relatively stable, an enzyme that has been poisoned by an irreversible inhibitor will remain inhibited even after the inhibitor is removed from the enzyme's active site. Irreversible inhibitors are usually very toxic substances. Lastly, we have regulation of enzyme activity. A cell that continually produces large amounts of an enzyme for which substrate concentration is always very low is wasting energy, and the production of the enzyme needs to be turned off. Now, a product of an enzyme catalyzed reaction that is present in plentiful, which is more than what the cell needs, amounts in a cell is a waste of energy if the enzyme continues to catalyze the reaction that produces the product. Thus, the enzyme also needs to be turned off. So, enzyme activity must be regulated to avoid wasting energy and to avoid wasting resources. Now, there are three mechanisms for the regulation of enzyme activity. We have the feedback control, which involves allosteric enzymes. We have the proteolytic enzymes and zymogens, and covalent modification. What is an allosteric enzyme? So allosteric enzymes are enzymes with two or more protein chains, usually in the quaternary structure, and two kinds of binding sites. So a binding site for a substrate and a binding site for a regulator. So allosteric enzymes are actually those enzymes inhibited by the reversible non-competitive inhibitors. Characteristics of allosteric enzymes all have quaternary structure. They have two kinds of binding sites, a binding site for substrate and a binding site for regulator, or what we call a regulatory site or allosteric site. Active and regulatory binding sites are distinct from each other in both location and shape. The binding of a molecule at the regulatory site will cause changes in the overall three-dimensional structure of the enzyme, including structural changes at the active site. Okay, so for allosteric enzymes, we have regulators. So these are the substances that bind at regulatory sites. Now, regulators can either be positive or negative. Positive regulators are those that increase enzyme activity. So, for example, we need large amounts of ATP at the moment, and what we have is not sufficient. Then the enzyme that will produce ATP must be activated. So what it does is that once it attaches to the regulatory site, it would alter the shape of the active site in a way that would be more susceptible for the substrate to attach. Now, negative regulators are those inhibitors or the non-competitive inhibitors. Feedback control is a process in which activation or inhibition of the first reaction in a reaction sequence is controlled by a product of the reaction sequence. Most biochemical processes within the cells take place in several steps rather than in a single step. As you can see from this, a different enzyme is actually required for each step of the process. Now, the process of each step or the product of each step is the substrate for the next enzyme. Now, feedback regulation of an enzyme occurs when a product of the reaction binds to an allosteric site on the enzyme and affects its catalytic activity. So, through feedback control, the cell responds to the amount of reaction product in order to regulate its further production. So, for example, in here, we have here the substrate. It attaches to enzyme 1 and it will be converted into an intermediate substrate. Now, enzyme 2 will not be able to catalyze the reaction unless the intermediate substrate A is produced. So, we have the negative feedback and the positive feedback. So, negative feedback results in inhibition of an enzyme, reducing activity of enzymes, or therefore stopping the pathway. Positive feedback results in activation of more enzymes, thus increasing the production of the product. Second, we have proteolytic enzymes and zymogens. Mechanism based on the production of enzymes in an inactive form. Proteolytic enzymes are enzymes that catalyze the breaking of peptide bonds that maintain the primary structure of a protein. So the inactive precursors are turned on at the appropriate time, which we call the zymogen, or the inactive precursor of a proteolytic enzyme. The activation of a zymogen will require an enzyme-controlled reaction that will remove some part of the zymogen structure. Now, this modification changes the overall three-dimensional structure of the zymogen, which will affect active site conformation. Last, we have covalent modification enzymes. When we say covalent modification, this is a process wherein enzyme activity is altered by covalently modifying the structure of the enzyme through attachment of a chemical group to or removal of a chemical group from a particular amino acid within the enzyme's structure. So meaning, certain parts of the amino acids that is building up the enzyme would be modified. So examples are phosphorylation and this dephosphorylation reaction. So this is the most commonly encountered type of covalent modification involving the addition and removal of phosphate group from an enzyme, which would usually involve the ATP molecule. So this two process is the off-on or the on-off switch for the enzyme.